Hi, today I'm going to talk about the primary, secondary, tertiary, and quaternary structures of proteins. So in the primary structure of proteins, it consists of a linear sequence of amino acids that is determined by the DNA. And the order of the amino acids determines the protein's folding and the protein's function. So the primary structure looks something like this. And also, even a single point substitution of an amino acid can cause a whole change in the function of a protein. For example, in sickle cell anemia, the amino acid glutamine is replaced by the amino acid valine. For the secondary structure, we have two types, okay? So number one, we have the alpha helix, which is just a coiled up spiral, and then number two, we have the beta pleated sheet, which is just two parallel strands connected by the hydrogen bonds between them. So the secondary structure, it provides local stability to the protein. These beta pleated sheets and these alpha helixes, how are they able to stay in that shape? Well, that is because of the stabilization provided by the hydrogen bonds between the COO group and the NH group. So remember what a peptide looks like. For the tertiary structure, it's when the amino acid chain, it folds into a unique 3D shape, and that is stabilized by four things. First, we have the disulfide bridges between the cysteines, hydrogen bonds, hydrophobic interactions between the nonpolar R groups, and we have the ionic bonds between the charged R groups. And the folding is assisted by chaperone proteins. So for the quaternary structure of proteins, it only exists when the protein has more than one polypeptide subunit. So basically, the quaternary structure, think of it as lots of tertiary structures together. So there can be two types of quaternary structures. We have the non-conjugated and the conjugated. So the non-conjugated protein only consists of polypeptides. So it's purely just made out of amino acids. So the conjugated proteins, that is when we have a polypeptide and a non-protein group together. So for example, we have a hemoglobin and we have the hemes. Now I also want to talk about the polar versus non-polar R groups. Polar R groups are hydrophilic, okay? They are water-loving, they have a high affinity towards water molecules. In a globular protein, they're going to face the water. So the hydrophobic region, they don't really like water, so they have a lower affinity towards water. The hydrophobic groups are just going to cluster together. That's going to give the protein stability. Think about membrane proteins, right? So membrane proteins, they have a lipid core, which is made out of hydrophobic regions, and outside they have their hydrophilic regions, which are facing the cytoplasm. Now, I also want to talk about fibrous versus globular proteins. So fibrous proteins, they are elongated, structural, and they are insoluble. For example, keratin and collagen. Globular proteins, they are very compact, functional, and they are soluble. For example, enzymes, or we have hemoglobin. Thank you so much for listening, and see you next time. Bye!